Hi there. In this lecture, I'd like to talk to you guys about nucleotides and nucleic acids. And we're keeping in with the theme of the idea that biochemistry is the chemistry influencing the biology. I'm going to talk to you about these nucleotides, which are these building blocks, and talk to you about the unique chemistry of the different nucleotides and nucleotides in general as compared to other small molecules like amino acids or just carbohydrates on their own, things like or lipids, for instance. Talk about the differences that you have with them and how the unique chemistry makes them all kind of the nucleotides fit together, get this larger thing, the nucleic acids, and then what that does and what that does biologically. So you guys are all kind of familiar with the idea that DNA is this storage of information, a storage of genes. And essentially, it's a huge polymer that's made up of lots of monomers in the same way as proteins are just monomers of amino acids to make this big polymer of the protein. DNA is just monomers, the monomers of the nucleotides. There are several different nucleotides. You guys are familiar with A, T, C, and G. There's also U as well with RNA. And we can break them down further into three different parts of the nucleotide. There's the phosphate, the sugar, and the base. We'll talk about those later. But aside from the nucleotides that make up the genes and the DNA and the genes, there's also other nucleotides. There's nucleotides in signaling and nucleotides in energy. For instance, you guys have heard of ATP, or ATP is just the adenine, but with just the T, triphosphate, with just extra phosphates. So here you can see adenine, and it would just be with an extra 2 phosphates attached to it. So there's other things like cyclic AMP, all sorts of things like that have a few different roles. But the most famous one, of course, is in DNA, which is just storage of information. So in this lecture, I want to cover nucleotides and nucleic acids. I'm going to talk about the structures of the common nucleotides. So remember I said it's made up of the 3 parts, the phosphate, the sugar, and the base. I'm going to talk about those and some of the common nucleotides, the A, T, C, and G, and really what differentiates between them. And then in knowing that, you can then know how they come together to form double-stranded DNA, how the chemistry influences the biology, how that chemistry influences them coming together, in a way that creates a kind of redundancy in the data, which is really important to make sure that there's no mutations and things like that. Very briefly, I will just talk about ribonucleic acids, but not too much on that. In general, I'm just going to focus on deoxyribonucleic acids. And then I'm going to talk about denaturation and annealing of DNA. The idea that if you have a double-stranded DNA and there's strand 1, strands 2, and they're interacting, strand 1 and 2, and they're interacting with each other to become kind of like one big molecule. The idea that this can be broken apart into 2 individual strands, and this is denaturation, but then it can come back together again. We call that annealing. And this underlies how PCR works. I'm going to talk about that. And then finally, I'm going to talk a little bit about DNA mutation and chemotherapy, because in chemotherapy, one of the main things is as the treatment of cancer, or really with a lot of with cancer, you have these rapidly dividing cells. You have a lot of rapidly dividing cells. You have a lot of DNA that's being replicated. And thus, one of the attractive treatments for cancer is to target DNA replication. Most of your cells are senescent and don't divide so much. The key ones that do divide a lot are, of course, the hair and the stomach. And that's why, in particular, chemotherapy also affect hair and stomach, also immune cells and things like that. But also chemotherapy can be used to target those things. Really, it targets anything rapidly dividing. And mutation, the idea that like this kind of comes in with chemotherapy as well. The idea that these nucleotides, nucleic acids can be modified and changed by the environment around them, and way that might affect the genes and affect the storage of data and information. So this is just really just a little bit of more in depth of what to expect coming up. the idea that nucleotides are a family of molecules. So it's not just in the storage of information of the ATCGU, but also, like I said, the transmission of information, such as things like cyclic AMP, and also for energy, ATP and NAD, NADP. But the most famous known is for the nucleotides as monomers to join together to form the polymers, which are nucleic acids. Nucleotides are not nucleic acid. It's when it's joined together to form the polymer. You guys are familiar with the most common nucleic acid, DNA, but there's also the RNA as well. And DNA stores information and it's transmitted as the mRNA. It's kind of like you have a recipe book, and inside your recipe book you have all of the different, which is your genome. Within that you have the DNA, and the DNA is the recipe book, and within that are the genes, which are just the different parts of the DNA, which are just different recipes. And you want to take out that recipe, kind of like the mRNA, and turn it into the actual kind of dish that you're cooking, which is the protein that you make. And tRNA and rRNA, that's the ribosomal RNA. These are also things that help out with the translation, but I don't really cover that. That's more for kind of a molecular genetics kind of course, but just really these belong to the RNA family, which is different from the slightly different from the structure of DNA, and we've covered that as well. So like I said, nucleotides are made up of three parts. There's the nucleobase, which is this heterocyclic ring, and it's heterocyclic because it's not just carbon, it also has nitrogen in there as well. There's two of them, the pyrimidines and the purines, and it's known as a base because it has these nitrogens in them, and we know that nitrogens often can act as proton acceptors, so have a kind of base quality to them. But these are between the different nucleotides. These are perhaps the most common changes, especially when it comes to the ATCG. In fact, these are the things that change. And like I said, we can divide them up into two families, the pyrimidines and the purines, which are modified slightly to create the different structures. And this is the structure of pyrimidine, and so the pyrimidine family, this but slightly modified. And this is purine, and the purine family are this but slightly modified. Now, if you get the base and you add it to the pentose sugar, and remember from carbohydrates as O's, so it's a sugar, pent, 5, so it's a 5-member sugar. If you take the base, add it to the pentose sugar, you get the nucleoside. That's 1 plus 2, not including the phosphate. And like I say, it's a sugar, it's a 5-membered sugar. Oh, I've drawn it wrong. It is a 5. Somebody let me fix it. It's a 5-membered sugar, like so. And like I say, it's a pentose. And then finally, to complete it and make it the nucleotides, not the nucleotide, till you add on the phosphate. The phosphate gets attached to the sugar at the 5' position. We'll talk a little bit more about that in a moment. So let's start breaking these down bit by bit into the 3 individual components: the phosphate, the base, the sugar. We'll start with the phosphate because it's the easiest one. Here's the phosphate. The phosphate is negatively charged at a neutral pH, like most phosphates, and it's typically attached to the 5' position of the sugar. So if we start with the sugar here is the anomeric carbon, carbon number 1. This is the one that used to be it was the carbonyl carbon. So it's carbon number 1, 2, 3, 4, 5, and we give it a little prime in nucleotides to differentiate it from the carbons of the base. So the carbons of the base are just 1, 2, 3, 4, 5, 6, etc. Whereas of the sugar, it's 1, 2, 3, 4, 5 prime each time. So we can tell the difference between them. And the phosphate is typically, especially when it comes to DNA, RNA, is attached to the 5 prime position. Now, when it comes to other nucleotides, it may be attached to different positions, but for the nucleotides that are going to make the nucleic acids, it's attached to the 5 prime position. Now, the pentose sugar, it's ribofuranose, or either ribofuranose in RNA or deoxyribofuranase, so it's been modified. It's an aldehyde sugar. Is that carbon number 1, 2, 3, 4, 5, which is going to cyclize to make ribofuranose, or beta, because the OH group of the anomeric carbon is pointing upwards. So it's beta, and it's as the sugars, all the sugars in life are all the D isomer, and it's the ribofuranose. So it's the ribose. The furanose is indicating that it's in the cyclic form. Notice that when we get to position 1, 2, 3, 4, 5, look at position number 2. This is in the ribofuranose. This is of RNA. Position number 2 has got the OH attached to it. But in deoxyribofuranose, which is for DNA, at position 1, 2, notice we've lost our OH. So this is the difference between DNA and RNA, is that the DNA, it's deoxyribose nucleic acid. So the nucleotides of deoxyribose nucleic acid are deoxy at position number 2, where it's lost its OH. Another thing is just a somewhat minor thing. This isn't so much at the so important at the undergrad level, but just to notice that this sugar actually has it isn't flat. It's not planar. Rather, it's got a slight puckering. This red here is that oxygen. You see that the carbon number 2, or 2' or 3' could be in endo or exo conformations. So it's not flat. Might have a slight kind of bend to it in the ring, and this will affect how the once the nucleotides chain together and form DNA, it will actually change the shape of the DNA and the way in which it will arrange. But that's not so so important at this level. But I just thought I might mention it. And then the third part, of course, is the nucleobase, and this is one that's really famous, the one that people know about. And like I said, they are derivatives of pyrimidine and purine. Pyrimidine, the longer, more complicated word, is the shorter, smaller, less complicated heterocyclic. It's got these 2 nitrogens in there, position 1 and 3. Whereas purine, the shorter, more simple word, is the more complicated base. It has a nitrogen at positions 1, 3, 7, and 9. And it's kind of this 6-membered ring and this 5-membered ring attached to it, which is kind of like the structure of, is the structure of purine. And like I said, both of these are modified to make the different bases. These are conversely are flat instead, or pretty flat. They also absorb UV light, and this is how we would measure DNA concentration in a research laboratory, or nucleotide concentration in a research laboratory. Is that you could use, put it in a spectrophotometer. You can shine UV light at usually 260 nanometers. Look at the absorbance of the light, and using Beer-Lambert law, we can figure out the concentration of the nucleotide in there, or the nucleic acid, or whatever it is that you're measuring. Because the more that it's going to be present, the more it's going to absorb, and so the less that's going to be detected by the detector on the other side. So it's shine through the light, and less is going to be detected on the other side. And so we can figure out from that what the concentration inside is. So this is the structure of the 2 purines. And like I said, they're based off the structure of purine here. The difference being is that for adenine, at position 6, you have this amine group. And for guanine, you at position 6, you have a carbonyl instead, but you have the an amine at position 2. Now, the way that this is going to change things is think about amine groups, think about carbonyl groups, amine groups. What do they do in terms of if they're in solution, if they're in an aqueous solution with water? What are they going to be doing? Well, they're going to be forming hydrogen bonds, and they're going to be forming different hydrogen bonds. They might be hydrogen bond acceptors. So, for instance, the lone pair of electrons on the carbonyl oxygen is a hydrogen bond acceptor, whereas actually the lone pair of electrons on the nitrogen is also a hydrogen bond acceptor, but in particular, the hydrogens are going to be hydrogen bond donors. So this this is kind of a little kind of hint about what's coming on, how the bases come together. It's the correct combination of acceptors and donors matching up between the bases. That's how they match up and they pair up. So that's adenine and guanine. The pyrimidines, there are 3 of them, because in DNA you have thymine, and in RNA you have uracil, and they're swapped between depending on which nucleic acid you have. There's not much difference between thymine and uracil. The difference is that at position 5, you have this methyl, this methyl group. If it's present, then it's thymine. If it's not present, then it's uracil. But both of them have the carbonyl at positions 2 and 4, position 2 and 4. Cytosine is the other pyrimidine. It still has the carbonyl at position 2, but instead it's got an amine group at position 4. Now notice again that these are 1, 2, 3, 4, whereas conversely in the sugars, I've done it again. Just ignore that there. In the sugars, it's 1, 2, 3, 4, 5, but it's prime, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And that's how, if I was to say, oh, carbon number 4, you know, I'm talking about this, not talking about this. If I was to say carbon number 4 prime, you know, I'm talking about this. That's how you, that's a kind of a little tool to help us tell the difference between them. Now, like I said, adenine and guanine are found in both RNA and DNA. These are the purine bases, and they are good hydrogen bond donors and acceptors. You've got the donor and acceptor group here. You've got the donor and acceptor group here, and I mean you've got them all over the nitrogens and the carbonyls. They all may or may not act as different hydrogen bond acceptors or donors depending on the conditions that they're at. And they're usually neutral molecules at pH 7. And the pyrimidines, just to recap, so this is the structure of pyrimidine. These are things which are based upon that, where they just have a change up here, either a carbonyl or an amine, and they all have the carbonyl at position number 2. But the difference between the thymine and the uracil, DNA and RNA respectively, is the presence of the methyl group at carbon number 5. And again, they got lots of hydrogen bond donors and acceptors, so they're going to be very soluble in water. But the exact hydrogen bond donors and acceptors really depends on that happen are very specific when they match up and pair for the DNA. And again, they're going to be neutral at pH 7. So how do we put all of these together? So you have, like I said, the base, and that goes down and attaches to the sugar, and then that then goes and attaches to the phosphate. So you have part 1, part 2, part 3. How do these all join together? So individually, so first of all, the base, pyrimidine or purine, attaches to the pentose sugar at position 1', exactly the same as if you had a polysaccharide, and it's just attaching at the anomeric carbon, reacting at the anomeric carbon to form, instead of the hemiacetal, the acetal. And it's a position at, but instead of it being an O glycosidic bond, it's an N glycosidic bond, because it's not an alcohol from a neighboring sugar, which is joining on to make the polysaccharide. Rather, it's a nitrogen from the base, so it's an N glycosidic bond. And of course, it's beta because the OH of the sugar is in the beta position. It's above. It's pointing upwards. So it's an N glycosidal bond onto a carbon 1' of the pentose sugar, and it's a phosphoester bond. It's a phosphoester bond to the 5' position of the pentose sugar. For the again, this is actually for the genetic nucleotides that are going to lead to some kind of nucleic acid. It may be attached at different positions, or maybe attached at 2 positions for different nucleotides, for non-coding non-genetic nucleotides. But for the genetic nucleotides, phosphoester bond at position 5' of the sugar. Now, to characterize this a little further, this is all kind of stuff that I mentioned, but just as to make it clear, the nucleotide is attached to the sugar at nitrogen 1 of the pyrimidines, nitrogen 9 of the purines, forming an N glycosidic bond. It's the anomeric carbon of the sugar, which is in the beta conformation, and it's in general it's pretty stable. It's fairly stable. It can be cleaved. So, for instance, if you had the sugar, the base, so it can be cleaved under certain conditions, especially in a kind of a low pH condition, but also certain mutations can cause it as well. For instance, UV radiation can cause base cleavage. So that's part of the reason why UV radiation from the sun is harmful and dangerous, and why we need to protect against us, because it will damage our DNA, for instance, causing depurination. So depurination, the loss of purine from our genetic material. And so our body needs to recognize that and repair that. Of course, the handy thing is that if you have your DNA double-stranded and it only matches up in the specific ways, so A is always going to match up with T. If something were to happen and you were to lose the T, you still have the phosphate and the sugar, but you've lost the T. And because of maybe some UV damage has caused some kind of caused hydrolysis, or there's been some kind of chemical environmental toxin, something like that, then it's broken off the base. Well, your body can go back and repair it, because it knows, well, here, this is A, which means this must be T. And so it can go in and repair and insert in a new T. This is quite important, but in general, it's fairly stable, and it's got to be. Otherwise, we would have life would have died out a long time ago, and life wouldn't exist as the way that it does. Now, just really some naming rules that you should be aware of. Now, actually, a lot of people in general, and myself included, get in the bad habit of calling the nucleotides by the name of their base, whereas actually, technically, that's actually just the name of the base. The nucleotide of adenine, once you add in the sugar, if you have adenine, once you add in the sugar, it becomes adenosine, and once you add on the phosphate, it becomes adenylate or adenylate. So here's the base, the base plus sugar, which is a nucleoside, the base plus sugar plus phosphate is a nucleotide, and they each have a different name. Adenine is when it's on its own. Adenosine is when it's with the sugar. Adenylate is when it's with the nucleotide. And you can do it exactly the same with the other ones. If you have adenine, but you have it with the deoxy sugar instead. So this would be actually RNA, which you can see here at the end. If you have it with the as it's DNA, it's with the deoxy sugar, there'll be the deoxy adenosine, and then it'll be deoxyadenylate for the DNA. Well, the same with, let's go to uracil. Uracil is the base on its own. It's uridine when it's with the sugar as the nucleoside. Notice that it doesn't have a deoxyuridine because uracil was always just RNA. So there is no DNA version of it. You could make it in the lab, I'm sure, but it doesn't exist as a in life. And then when it's the nucleotides, once you've added on the base, it's uridylate as the RNA. On the other hand, thymine, deoxythymine, deoxythymidine. Thymidine doesn't really exist in nature, and so it'll be deoxythymidylate of the DNA. So it's easy to make the mix up and just call the nucleotide guanine. That's actually just the name of the base. The nucleotide is guanidinylate. And this is just really showing the whole structures of them. Here we have the DNA. So this is the deoxyribonucleotides. These are the nucleotides that make up DNA. Also sometimes called deoxyribonucleoside 5-monophosphate, because here in this kind of peachy color is the deoxynucleoside. And then remember 1, 2, 3, 4, 5 prime phosphate. So deoxyribonucleoside 5' monophosphate. The monophosphate being added there. Or it could be 5' triphosphate, for instance, ATP. So that would be the adenylate triphosphate. And so there are the deoxyribonucleotides. There's the adenylate, the guanidine-based, thymidine-based, and cytidine-based deoxy with the deoxy sugar. It's missing its OH. On the other hand, the ribonucleotides, here's the base, here's the sugar, it's got its OH, so it's just a ribonucleotide, and there's the phosphate of the adenine, guanine, uridine, cytidine. Now, this is getting all of these names can be difficult, and this is a lot. And I've been talking about these and showing you all of these different things, and it's probably very, very overwhelming. They'll just be like, how all of these subtle differences that's so big, but it's so subtle, and it's just so overwhelming. So let's just take a breath, breather, break it down, recap. What are we looking at? Well, first of all, we know that you have the sugar and the base. The main thing really is to take away that the base is attached to the sugar at position 1', which is the anomeric carbon. It's the carbon that used to be the carbonyl carbon. It's the one that was the aldehyde carbon in this case, and they're joined here by this N-glycosidic bond. Then if we go around 2, 3', 4', 4', 5', and then at the 5' position, that's where the phosphate is going to add on. And this is by the phosphoester bond. So breaking down to the 3 bits: the base, the sugar, the phosphate. The base is attached to the sugar, anomeric carbon, by an N-glycosidic bond. The phosphate is attached to the sugar at the 5' position by a phosphoester bond. So once you got that, that's the first part. The next part is to recognize in the sugars, where you have at the 2' position, you either have the oxy or the deoxy. So if I was to show you guys a sugar, I can show you guys, let's say, this one and this one, and this is the ribofuran. Oh, and this one is the deoxyribofuran. So oxy and deoxy. And often we don't even bother with the oxy, but you can if you like. But the main thing is that this is deoxy. That's super important. Then if you wanted to memorize the structure of the nucleotides, you've got the, first you've got the 3 that join together, the bonds that join them together, knowing, just look at position 2', is it an oxy or a deoxy? That'll tell you whether or not it's DNA or RNA. Then if you wanted to memorize them, you can learn the 5 bases. And the best way of doing that is to just get familiar with the structure of purine and pyrimidine. Start out with pyrimidine. That's just a 6-membered ring, and it's just the nitrogens at position 1 and 3. And of course, the nitrogen is going to be at position 1. I mean, think about naming rules of these kind of heterocycles. The nitrogen is always going to be position 1, and and it's always going to be numbered by the next nitrogen along, which is always going to be the smallest number. So it's 1, 2, 3. So that's the structure of pyrimidine. And then purine is similar. You have this 1, 2, 3, 4, 5, 6, this heterocycle. And then at positions 4 and 6, you've got another 5-membered ring coming off. And it's positions 7 and 9, which are opposite each other, which are going to be the nitrogens as well. So if we go for the pyrimidines, if we just draw our aromatic ring, and wherever you put position 1, you could put it here, here, wherever you want to put it, just as long as it's, you just call that one position 1, and then 1, 2, 3. So there's position, there's 1 and 3. And so this is going to be the pyrimidine. And then for the purine, it's a similar kind of thing, except you want to do this extra ring on there, and you want to, as 1 and 3, 4, positions 4 and 5 are these ones here, position 4, position 5, and the nitrogen is going to be opposite them with position 8 in between. And then once you've got the structures of that's the purine and the pyrimidine, once you've got these ones kind of committed to memory, then you can learn the differences between them. So adenine, I remember adenine, A for amino, so it's just got an amino group, and it's just attached here at the top, the amine group. Whereas conversely, I just say guanine. When I say the guanine, I remember guanine is the other one. So if you think of the structure of purine, and then you want to say, well, adenine amino, that's just got the one amino group, so I'm just going to put the solo amino group. And then guanine, I say, well, that's the different one. So because it's different, I'm going to swap out the amino group for a carbonyl. You're going to add an amino group down here instead. So it's just that different one. You just kind of swap it and put it into a different place. Pyrimidine, you just remember of the ring. It's always got, they've all got the carbonyl here, down here. And then cytosine, I say, well, cytosine is C, is early in the alphabet. So I say that it's early, so it's got the amine up here. And then if you want to swap it out for the other 2 bases, then you just exchange that for the oxygen to make it the carbonyl. And then the difference, and then so that will give you the T and the U. And remember, the only difference between the T and the U is the methyl group. Uracil doesn't have a methyl group. Thymidine does. So that's just some little rules if you wanted to memorize the 5 bases that make up genetic nucleotides. So one thing that I did want to just briefly mention is just how these get modified as well. So, excuse me, you notice that, for instance, this is cytosine, and cytosine is not supposed to. If we go back to, here we go. So remember, cytosine is the one that has the amine, is the pyrimidine that has the amine group at the top there. So here we go. It's got the amine group at the top there. Remember, your thymine has got the it's got a methyl group, but it's got the carbonyl at the top. This doesn't have the carbonyl at the top. It's got the amine at the group at the top. So it's the cytosine. It's C, but it's got the methyl group. So this has been you think so what am I talking about here? What I'm talking about is that the bases can be modified. So the ATCGU, those are the basic units. But sometimes you're in biology, we want to change them and mess around with them a little bit, because that can help us store even more data, right? Rather than just having, is it an A or is it a C? How about having a methyl or a C methyl? It's a way of just kind of changing things a little bit to add a little bit more information. And what will happen sometimes is that the body will methylate some of the bases in order to modify them slightly. So instead of it being cytosine, it's 5-methyl cytosine. And this can the body can detect this modification and say, ah, this is cytosine, but this is different. This is methyl cytosine. It can recognize that it's kind of the same, but slightly different. And that can be used to communicate a slightly different thing. For instance, if you wanted to, it's often used as considered in genetics as an epigenetic marker. It's something that's kind of like a post-translational modification of a protein. It's kind of like a post kind of genetic marker in some kind of way, where you have your genes and you can't modify your DNA. Or, I mean, you can, but that's a whole different kind of beast that's relatively new in research as well. But in general, DNA, genes don't get modified that much. It's fairly kind of fairly solid. And again, this is a kind of work in progress in research and our understanding. The picture's a lot more complex. But what we can make kind of like soft changes. And these soft changes are putting on methyl groups, taking off methyl groups. And for instance, some genes get methylated to switch them on, and some or get the methylation taken off to switch them back off. Or methylation can switch genes on or off by putting methylation into the bases of that particular gene. If I have a gene ACCG ACC, and normally your machinery is going to come along and transcribe it and translate it and turn it into a protein that does something. Well, maybe you got methyl cytosine. There's the methyl group. And so the machinery will come across and it'll get to that methyl and go, nope. And so it won't transcribe it. And so it's a way of gene, can be a way of gene regulation. Various bases can be methylated. For instance, 5-methyl cytosine, so the cytosine gets methylated at the 5 position. Or this amine could be turned into a secondary amine in adenosine. Guanine, secondary amine. Hydroxymethyl cytosine. So it's just different groups can be added to the bases in order to modify the DNA slightly, kind of as a soft edit. And for instance, human DNA is pretty, and mammalian is eukaryotic actually really in general is pretty methylated. And so if something comes along that isn't methylated, as a very undermethylated, actually our immune system and the inflammatory immune system inside your cells can say, hey, this genetic material is really undermethylated. Something's not right here, and it can therefore recognize it's being foreign, and then can degrade it. So for instance, like viruses and things like that can stop you from being genetically modified. Or for instance, like I said, it's a way of regulating your genes, switching methylation on and off. Sometimes methylation switches it on, sometimes it switches it off. Something really, really super interesting is that these epigenetic markers can be inherited. So for instance, there was a study that was done with mice where pregnant mice were vastly overfed and like they're really, really overfed and made kind of diabetic, huge amounts of sugar. And so what happens that between the mother to the progeny, the child, the mother mouse to the child rat progeny, they had the expected kind of DNA and genes, and perhaps the gene for glucose metabolism maybe is very, very well conserved. And so the conserved unchanged. So maybe the genetic coding, ATCG, between the mother to child was largely conserved, wasn't changed very much. But what happened was that when the mother was really, really overfed, and so there was a different kind of epigenetic change, different change of the methylation markers on the mother's gene to maybe switch the gene on or off in order to respond to the influx of food. That was carried across to the child. And so, these kind of, and this can happen to all kinds of things. That was an example that was given with a classic example with food, but also it's believed it may happen with trauma as well. So for instance, and this is, part of how generational trauma gets passed through, literally can be passed through genetics, where perhaps the gene may not be changed that much in terms of the raw coding ATCG. But when you become kind of extreme mental stress can cause a epigenetic modification of your coding. So certain methylation patterns that are responses to certain stresses and all kinds of things like that. And that can be passed on to the children, where the mother and the child may have a similar genetic coding and the same genetic coding that's in the whole of the population. But if the mother received trauma, like a mental trauma, then this can be passed on to the child by the methylation pattern being passed along, which is super interesting as well. So I just thought I'd mention that. This is just one way in which DNA can be modified that isn't necessarily considered a kind of often a kind of mutation in the classical kind of sense as it's often talked about, or particularly damaging. It's usually an environmental response. And this is the idea that, again, the environment is causing a biological response. That biological response is causing a chemical change. That chemical change is then causing a new biological response. The biology influences the chemistry influences the biology. This is what biochemistry is. And then just to mention as well, so just some more modifications. So we talked about bases and base modification with that methylation, but just the idea with the phosphorylation pattern. So I mentioned that the sugar is our base, sugar, and phosphate. And notice it's at the 2' position instead of the 5'. Here it is at the 5'. 1, 2, 3, 4, 5'. So adenosine 5 monophosphate. So this is adenosine 5 monophosphate. So 5'. I keep forgetting the prime. Adenosine 5' monophosphate. So this is the just the normal A nucleotide for that you might have in genetic material, but also serves the same thing as its AMP, is in the same AMP when we're talking about energy. And if we were to stick 2 more phosphates on it, it would be ATP, adenosine 5' triphosphate. That's ATP, which is the storage of energy. And it can be ADP as well, of course, which is when it was just 2 phosphates. So this is what's happening. It's the same thing. AMP is the same thing as the genetic A in the genetic code, the same genetic letter. And ATP is just sticking on extra phosphates. But you can also change the position of that phosphate, put it at position 2, put it at position 3, And this can be instead for communicating, for instance, with the ATP, that's for energy. These ones can be for information as instead. And the classic example is cyclic AMP, C-cyclic AMP. And this is where the phosphate is kind of forming a cyclic kind of thing here. It's attached to the 2' and the 3' position. And this is a signaler. This is linked to receptors on the cell and increases and is released and increases in the cell in order to signal when the cell was stimulated by an external signal. You can also have it with cyclic GMP, which is instead of it being adenine base, it's a guanine base. So it's the it's not just these nucleotides are not just for genetic material. They could be for energy. They could be for signaling, and often that's to do with the positioning or the number of the phosphates. Okay. Now, like I said, with the genetic material, these are that's the monomers are the nucleotides, and they join together with other monomer nucleotides in order to make the polymer of the genetic material, the nucleic acids of DNA or RNA. Now, recall that the of the sugar, the phosphate is attached to the 5' position by a phosphoester bond. Now, the this phosphate is then attached to the 3' position of another sugar. Kind of there. Well, it's going to be perhaps a little bit easier to show. I drew the 3' underneath, and it's going to be difficult to draw the sugar like that now. It should have been above, and now my text is in the way. But the 3' position, we have here the 1, 2, 3, 4, 5. Then there's the phosphate attached to the 5' position, and then if we had another sugar, 1, 2, 3, it will be attached to the next one along at the 3' position of the next sugar along, and then this then forms. You got 1 phosphoester bond, 2 phosphoester bond. So this is now known as a phosphodiester bond. So I'll try again on this one here. This is the RNA molecule. It's the RNA because it's got the oxy at position number 2', whereas this is the DNA. So the 2' position is the deoxy. So we start with this base, 1, 2, 3, 4, 5. 5'. There's the phosphoester bond, and the next sugar along, or rather the sugar, I guess in a roundabout way, the sugar before it, 1', 2', 3'. At the 3' position is where it's attached to the phosphate, and now you've got 1, 2 phosphoester bonds, so it's a phosphodiester bond. Now we describe DNA and RNA as going from the 5' to 3', and that is because this is in the sequence. This would go UGC, UGC, and notice, and it goes, so it's going down like this. Now notice that the exposed end, the one that's free, is the one which there's nothing attached to the 5'. I mean, there's the phosphate, but there's nothing beyond that. So this is known as this is the end, and the end is at the 5'. So this is the first sugar. There's nothing before it, so its 5' is free, so it's the 5' end. Whereas if we go down to the last one in the sequence, the 5' is bound to something, so it's not free. But the 3' is not bound to anything because there's nothing after it. So we consider that this the 3' end. So the ends are dictated by whichever group of the sugar is free, And the first one in the sequence is is always the 5', and the last one in the sequence is the 3'. So it runs 5' to 3'. And this really kind of summarizes it, between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of the next nucleotide. That's how this is formed. We have the between the 5' of one and the 3' of the previous, exposing the 3' and making it so that the first is the 5'. That's free. So you guys may be familiar with the idea that RNA is less stable than DNA. And there's been this talk about there it is. There's been this talk about the oxy and the deoxy and what that means necessarily. And that is the idea that RNA is less stable than DNA because of the hydroxy group at the 2' position can be attacked by a base. And when it's attacked by a base, it will cause a chain reaction where this OH will abstract the proton from the hydroxy group at position 2' of RNA. This then can then attack the phosphate group of the in the linkage. So if we have our RNA 1, RNA 2, and they are linked by a phosphodiester bond, and you have the OH of RNA 1, which is attacked by base, and then this will then attack the phosphate. And then this will then cause the phosphate will then break its bond, break the bond with the next nucleotide, and form a bond with itself, and cause a cyclization. So as you can kind of between RNA base 1 and RNA base 2, a kind of a base may come along, something like sodium hydroxide, abstract that proton. The oxygen of the attached to the 2' position will then attack the phosphate. That phosphate will then break the bond with the phosphodiester bond with the base after it, RNA 2. And instead become bonded to the oxygen, forming a cyclization and breaking the bond with the phosphate. And here you can see where it's kind of cyclized, but broken the bond is base 1, base 2, broken the bond with the one after it. So if you have a chain of RNA, so one of these circles is an RNA, and if base comes along, it will attack, and then this will then kind of cyclize with itself and break the bond, cause a cleavage in the RNA, and then you get degradation of RNA. So RNA is unstable at basic pHs because the base will cause a chain reaction breakage in the RNA, because the RNA will just kind of the phosphodiester bond will break and you just get a cyclic derivative instead. But this doesn't happen with DNA. I haven't got an example, unfortunately. It doesn't happen with DNA because instead of being the OH, it is the H, deoxy, attached to the 2' carbon. And so the base cannot attack as it can with the DNA. And so you don't have that same kind of chemical reaction. So DNA is a lot more stable. DNA is more stable. RNA is less stable due to the presence of the 2' hydroxy group. That's why, for instance, we can take a prehistoric ice get the DNA of a woolly mammoth and then use that to clone a woolly mammoth. When we're doing research, we often store nucleic acids at a slightly acidic pH, for instance, pH 6.0. We might put it in some tris buffer, pH 6.0. And we do that because the DNA will eventually degrade a little bit, but especially RNA will degrade. So we just lower the pH slightly in order to decrease the extent of this chemical reaction occurring. But of course, DNA and RNA, oh, that should be a D. DNA and RNA can be degraded by their respective ASEs, DNAs, RNA, enzymes which are expressed in order to break down nucleic acids because the body, the cell, needs to do that. Now we'll kind of switch gears and talk about how these join up with each other and how they how the bases complement each other. And I think I'll stop here and I'll turn it into a fresh video as well.